Let's just get started.
No, don't say anything.
Okay.
Yeah, let's go.
Morning, everyone.
Hey, Carolina.
Hi.
Hi, Carolina.
Nice background, Rob.
Thank you. We'll see if you can figure out what it is. Wow.
It's trees. They're trees. I figured it out.
Yeah, who shall speak for the trees? That's what I want to know.
The ruffalo trees. The trees, the trees, the ruffalo trees. Oh, man.
Hello.
Yana, you're being recorded. Bonjour.
Bonjour.
Thomas.
Okay. I grew up in communism.
Yeah.
Guess you were being recorded all the time. I have no issues with Big Brother. Big Brother's good. Jennifer, how you doing? She's muted.
Muted.
You're muted, Jennifer.
You're muted.
There you go.
But it's fun to try to guess what Jennifer's saying, though.
It's just great.
Huh.
Wow, that was quite an endorsement. All right, well, good morning, everybody. We'll get started in maybe, I don't know, 30 seconds or something.
You're looking in the horizon, Rob. What did you find? I like this view, looking to your right, far in the distant sky.
Yeah, it's really nice.
It's super nice.
Aaron, I can tell you about it at some point.
Thomas, are you a PSG fan at all?
Sorry?
Are you going to watch the game tomorrow?
Tonight. I think it's tonight.
Oh,
No?
tomorrow night.
Okay, sorry. Yes, I am. Not that I'm a fan of football, but my son is, so I have to.
All right, that sounds like a good time for us to get started. So I'm going to get started. So good morning, everybody. So I'm really delighted to be here today to close out this year's Physical Biology of the Cell lectures. And my idea really is to try and put a bow on the gift that we've been given over these last few weeks. And I like the use of the word few, and in celebration, I'm wearing my PBOC shirt from last year. For those of you who haven't seen it, this has got the scale bar with few on it, which I think is delightful. So the talk is entitled Schrödinger's What is Life at 75? The Physical Aspect of the Living Cell Revisited. And I guess I want to alert you that ahead of time that as often I do, I'm probably going to say a number of things that are philosophical. So, you know, if you're in the mood to hear a data-only talk, this might be a good time to hang up. So before I begin, I guess I wanted to just say to the alums of the course how much you've meant to all of us and what a privilege it is to be part of this community. So this is the 4 years of the course. Many of you will recognize yourselves. I would like to just point out there's some weird stuff going on with Manuel sometimes, like in this one. I don't know what's up with that. And then, you know, I think Phil Nelson must be the only person ever to actually wear a coat to the MBL beach, but there you go. So anyway, thanks to all of you. Thanks to Wilson. And so, yeah, that's what I wanted to say. So before we begin, I guess I just want us all to take 5 seconds, just to take a pause and reflect just for a second on the title of Schrödinger's book. What is life? People have been thinking about that forever. Steve Quake just sent me a mail this week with the claim, it was an argument that the greatest biologist in history is actually Aristotle, which I find a little weird, but that's an interesting take. People have been thinking about this question for a long, long time. And I guess I want to say that, you know, it's very easy to look at the work from previous generations and to feel haughty and to feel as though, oh, well, that was quaint, but they didn't really know what they were talking about. And I hope that, living through the coronavirus pandemic has reminded even us as scientists the need for humility. The antibody testing fiasco should just be a sufficient reminder. But I guess I wanted to call your attention to some great successes, but great confusions also over history. So in the 1770s, Corti had already discovered what we now call cytoplasmic streaming. And the reason I'm bringing this up is because the notion of what is life has been bugging people. And every time they think they maybe have a little bit of a handle on it, something new shows up that just doesn't make any sense. And cytoplasmic streaming is the kind of thing that could lead you to become a vitalist, because you're like, well, where does that energy come from? And then Thomas and I were talking this morning about this amazing book, the thing I show you in the middle, by Tremblay. And the reason that this is so crazy is that this guy, Tremblay, was basically an au pair, it turns out, in Holland at this estate. And he happened to discover Hydra. And I kid you not, it was like the discovery of CRISPR in our era. Everybody wanted it. So he ended up, he lamented in his letters how he had become like nothing more than a source to provide people Hydra, because they were so blown away by the fact that you could chop this organism up into 50 different parts and it would just come back and become a Hydra again. And then, the debate between Galvani and Volta over the nature of animal electricity. And, is animal electricity the same thing as the electricity that was being discovered by Volta in the context of the voltaic pile, or Benjamin Franklin, and so on. And, the fact that you can, if you think about it, the fact that a tardigrade can be frozen for 30 years, or that whole fertile plants can be frozen for 30,000 years in the permafrost, is shown in that PNAS paper. And yet, in the end, they're alive. That should show us some of the difficulty of the question. And I also wanted to point out that no less a luminary than Thomas Henry Huxley got so confused. So if you have the moment, go search on Bathybius Heclei. It was the outcome of the Great Challenger expedition. The Challenger expedition was a 3-year voyage of the ship that I show you here, and you can look up the log and letters of the Challenger. And one of the things that they discovered on the bottom of the ocean floor was some sort of a jelly substance that wasn't jellyfish. And it was just weird. And in the end, Huxley went off on this total tangent having to do with a new form of life, a new idea about the organic matter. And it ended up being inorganic, it's confusing. So anyway, that's just kind of by way of introduction to the title. So the talk outline goes like this. I'm going to start out with the verdict of history on the book, which I'm going to claim is premature. And then I'll use chapters 1 through 5 to talk about the nature of heredity and some of the great outstanding challenges in those questions to this day. And then the last bit, we'll talk about Schrödinger's very controversial use of the word negative entropy. So the circumstances surrounding the book. So Schrödinger had to leave Austria, his position as a professor there. He was very distinguished, obviously. We're talking something like 1938. And he had to leave in a hurry and basically had nowhere to go. And it turned out that the Irish Prime Minister, Eamon de Valera, had started the Irish Institute for Theoretical Studies or something like that. I don't think I remember the name properly. And he was absolutely thrilled. Can you imagine the Prime Minister of a country being thrilled to be able to attract Erwin Schrödinger to come and be the first theoretical physicist at his place? And so Schrödinger spent 17 years there. And of those times, he referred to them as the happiest years of his life. And while he was there, he was asked to give a series of public lectures, and he chose the subject of what is life. At the same time, he wrote this amazing book called Statistical Thermodynamics, and that's very important. And it's illustrated by the first of the quotes. "No perception in physics has ever seemed more important to me than that of Boltzmann, despite Planck and Einstein." So, it's important for you to realize he grew up in Austria, and he went to the university where Boltzmann was a professor only 2 years after Boltzmann's tragic suicide, and always lamented the fact that he was not able to have Boltzmann as his teacher. But he did have Boltzmann's main protege, Fritz Hassenhorl, and look what he has to say about that. "No other human being had a greater influence on me than Fritz Hassenhorl, except perhaps my father, Rudolf. He took 5 courses over 4 years from Hassenhorl, He said it influenced me more than anything else. So fundamentally, Schrödinger was a statistical physicist first, foremost, and always. And because of his relationship with his father, he was an only child. He grew up in a highly intellectual family. He was an absolute mastermind and a polyglot. He knew Latin, he knew Greek, he knew Spanish perfectly, spoke English sans accent. I mean, he was just a phenom. And with his father, he had this amazing relationship. His father was basically a botanist. And so although people love to trash Schrödinger as though this is some sort of an amateurish effort, in fact, he had a lifelong interest in biology that was very cultivated. Note what he says. Later on, when he had taken up botany, and I had virtually devoured, speaking of his father, when he had taken up botany, and I had virtually devoured The Origin of Species, our discussions took on a different character. Of course, I soon became an ardent follower of Darwinism, and still am today. So biology was a lifelong devotion for Schrödinger. And when I say let's have some context, I'm anticipating the growing rage that I feel towards what Perutz had to say about this book. So the usual story. So Schrödinger had a huge impact on many of the early luminaries of molecular biology. Crick said it suggested that biological problems could be thought about in physical terms, and thus it gave the impression that exciting things in this field were not far off. Some of you may not know, but Seymour Benzer had a PhD in physics. He was a physics professor at Purdue University, and he read Schrödinger's book, and that led him to basically retool to learn how to do phage. He said, I had always had a latent interest in biology. It was particularly Schrödinger's book that turned me on, Seymour Benzer. So this is very cool. This is a letter that Crick wrote to Schrödinger. Note the date, 1953, August 12th. So this is 3 months after the paper came out on the double helix. And he says, Dear Professor Schrödinger, Watson and I were once discussing how we came to enter the field of molecular biology, and we discovered that we had both been influenced by your little book, What is Life? We thought you might be interested in the enclosed reprints. You will see that it looks as though your term aperiodic crystal is going to be a very apt one. Yours sincerely, Francis Crick, FEC Crick. And at the top, notice, Department of Physics. Kind of cool. So, so not all recipients of this book were positive. So Max Perutz says, sadly, however, a close study of his book and of the related literature has shown me that what was true in his book was not original, and most of what was original was known not to be true even when the book was written. The apparent contradiction between life and the statistical laws of physics can be resolved by invoking a science largely ignored by Schrödinger. That science is chemistry. Sorry, you know, you guys should check out Perutz's autobiography. It says, I wish I'd made you mad earlier, or something like that. That's the title. Well, yep, he succeeded. Schrödinger's discussion of thermodynamics is vague and superficial to an extent that should not be tolerated, even in a popular lecture. That's appalling. You know, I have a good friend, I'm not going to say who's a professor of physics somewhere that teaches stat mech, and he was telling me recently that he shares my view, which is that the treatment of stat mech actually in What is Life is one of the best you'll ever see. It's not based on equations, but it's really beautifully done. So, you know, people had negative reactions. Fair enough. So what's the big question that he poses? And this is something, you know, that you probably see quoted now and then. He says, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? So my spin on the book, the whole thing turns on these two words, account for. And indeed, I would claim that that's the whole point of the physical biology of the cell course, to show examples of such accounting. So let me give you an example from physics, and it has to do with Schrödinger himself. So we're talking about 100 years between the discovery of spectral lines and Niels Bohr's first, let's call it juvenile attempt to actually understand the formula that characterizes the wavelengths of spectral lines, which is shown in the lower left. That's the Balmer formula, discovered by a mathematician named Johann Jacob Balmer, who spent his whole career as a teacher at a girls' school, high school, mathematics. But he was interested in the regularities of the spectra, and he worked out this formula. And in fact, he made a prediction about a spectral line that he did not know about, which was actually there, which is cool. And Schrödinger, in his early papers, 1926, came up with a formula that's basically the Bohr formula. And, you know, I want your minds to be blown. The Rydberg constant, which was originally a phenomenological parameter, which you can today, right now, take a screenshot of this, use the numbers that I have up there, and for yourself, work out the Rydberg constant. It'll work perfectly. All you have to do is take the difference between two of the lines and you'll succeed. So, but what's stunning is that the Rydberg constant ends up being a combination of the mass of the electron, the charge of the electron, and Planck's constant. You know, who would have thought? Here's what Planck has to say about spectral lines. If the question concerning the nature of white light may thus be regarded as being solved, the answer to a closely related but no less important question, the question concerning the nature of light of the spectral lines, seems to belong among the most difficult and complicated problems which have ever been posed in optics or electrodynamics. And so, Schrödinger, when he talks about accounting for, what he means is that you actually can make predictions about what's going on with a natural phenomenon, and in this case, it's spectral lines. And I just want to say, note, naming and classifying the spectral lines was no more accounting than is identifying genes and pathways. Good, we know the names of pathways and we know the names of proteins. That's great. I applaud it. I am super impressed. But sorry, that is not be able to account for a phenomenon. And, as I said, in the 1800s, there were huge tables. And many of you probably know, we had the Balmer series and the Passion series and other, there's other series of spectral lines for hydrogen and then other elements as well. And you can see this on the table on the left, or the book. That's 100,000 different spectral lines. So, yes.
Manuel is asking, was Balmer's educated guess inspired by the Newtonian paradigm with inverse squared laws?
No,
how did they guess
that
functional form?
No, not that I'm aware of. No, I think it was, it was strict empiricism. He just sat there and played around with it. And, actually, Manuel, it'd be fun for us to dig into that. But, one thing that you could really enjoy, and actually Nico did this and probably didn't tell you, but he and I read this paper from Balmer a few months back, which is absolutely amazing. I mean, I recommend all of you. It's a one-pager. But there's nothing mentioned about Newton and inverse squares. That's a great, really good question. So there's this great paper by Kirshner, Gerhardt, and Tim Mitchison. And I just want to comment again on this notion of accounting. So this is them in the year 2000 at the turn of the millennium, speaking about the current state of the art in biology. And I love their playful title, Molecular Vitalism. At the close of the 20th century, genetics reigns triumphant as a central theme in biological thought. The sequence of the human genome is widely seen as the starting point for biological investigation in the next century, and the debate on the origin of life largely defines alive as equivalent to accurately transmitting a genetic blueprint. We do not question the importance of genetics nor dispute the role of DNA as the blueprint for all the components of living systems, but we think it worth asking to what extent the post-genomic view of modern biology would convince a 19th century vitalist that the nature of life was now understood. How close are we to understanding how a single cell functions or how an embryo develops? If the answer is not so close, will true understanding of living systems come from further annotating the database of genes, or must we explore the physical chemical nature of living systems? I won't read more, but you should go read it. So my simple thesis is this: the time has come to deliver on Schrödinger's call for a physical basis of the living organism. And what I want to do is tell you two little vignettes showing what I would call our little steps for little feet approach to precisely the questions raised by Schrödinger in chapters 1 through 5, which is primarily about the genome, and chapter 6, which is about living matter. Before I do that, I want to take this opportunity, though, to say that, in some sense, my real mission as the last speaker for the course, and the reason that I decided to be the last speaker, was that I knew I would have this opportunity to look back upon the last 3 weeks. And I just want to point out, we started very strong with Jeremy Gunawardena pointing out to us the need for including the expenditure of energy to understand what's going on in living matter. Down at the lower right, we had this really fun treatment from Maha about termite mounds. And what I liked there was, he invoked in some sense the PDEs of physics to account for things like gas and temperature and so on. And then across the diagonal, if we're playing tic-tac-toe, which I win now, we have JLS and Amy and Steph. And, they showed us really creative and interesting ways to think about the notion of phase separation. The top two were quite surprising. It was really fun to hear about coronavirus and HIV from that point of view. And then Steph with respect to bacterial transcription. So, I think that really more or less every single speaker gave an example of accounting as I would like you to think of it, as I think Schrödinger meant it. So chapters 1 through 5, the nature of heredity. So what Schrödinger was up to was he was interested in a question of whether or not the laws of physics as he knew them. And now I want you to just hearken back to the fact that he's from Vienna. He went to the university where Boltzmann was. He was a statistical physicist. So that was what it was in his mind. And he wanted to know you know, do the things I know from statistical physics, how do they square with what I know about the transmission of genetic information? And one of the questions he wanted to answer was, how big is the gene? And he means by gene, you know, how many atoms is it built up of? And the reason he cared about that is he knew that there would be fluctuations for small numbers if it was traditional statistical physics. I'm not going to go into detail here. I'm just going to tell you he did 3 different really interesting estimates of the size of a gene. The top one is very easy for you to do. It's wrong in some sense, but it's cool, which is he thought that the banded patterns in chromosomes, it wasn't his idea. Others thought that the banded pattern in chromosomes, that each band corresponded to one gene. So it's very easy to do an estimate of what the size of a gene is. And you'll find it's too large, actually. The next idea is good. It's probably good enough, but the mapping had been incomplete. You'll probably remember that Thomas Hunt Morgan and Sturtevant had used the crossing, basically genetic crosses, to look at the proximity of genes. And so if that was a complete map, this probably would have worked reasonably well. But the brilliant idea was basically this famous 3-man paper that Delbruck was part of, where they looked at the rate of mutation induced by X-rays, and they knew the dose of X-rays because they could get the rate of mutation in air, meaning the rate of ionization of air. And from that, they could figure out, based on a given intensity, how many collisions they would expect with the genetic material. And so out of that came a reasonable estimate of the number of atoms. And let's say it's something on the order of 1,000. And so what bugged Schrödinger was he thought, well, this is going to lead to statistical fluctuations. There's no reason that we should imagine, you know, Thomas mentioned this yesterday. It's a really interesting part of the book where he talks about the so-called Habsburg lip. So he was intrigued by the fact that the Habsburg lip was robust over time. It survived from one generation to the next. And so he wanted to know, you know, like, what would that mean at the level of atoms, which is such a cool question. Schrödinger says, however, it needs no poetical imagination, but only clear and sober scientific reflection to recognize that we are here obviously faced with events whose regular and lawful unfolding is guided by a mechanism entirely different from the probability mechanism of physics. You know, I'm sorry, Perutz was just being snide when he belittles what Schrödinger was up to. And I also think it's very interesting, you know, this is 20 years after, less than 20 years after Schrödinger created the wave equation that bears his name, you know, really revolutionized physics forever. And yet, here's what he has to say. These facts are easily the most interesting that science has revealed in our day. So his point had to do with fluctuations, okay? I'm not going to go into this. It has to do with the fact that there's 1 over root n fluctuations. And, you know, this has become a biological theme par excellence. I mean, when we think about the segregation of proteins during cell division, it's a perfect place to be thinking about this sort of coin flipping mentality. And, you know, I don't know if Avi's on the call or not, but his group at Berkeley, they've done a beautiful job of thinking about segregation of carboxysomes and show that there is an active mechanism to actually prevent these 1 over root n fluctuations. So the vignette I'm going to tell you about is primarily the work of Nathan Beliveau and Susie Beeler and Bill Ireland, and it has to do with our attempt to try and understand something about the genome, you know, in modern terms. And it all comes down to a puzzle. You know, Janne mentioned the other day he likes puzzles. I don't like them that much, but I do think this is a funny puzzle, which is if I were to send out this particular sequence out into the world and ask what it means, basically nobody would be able to tell me. It's only 100 letters or something from E. coli. It's pretty pathetic, if you ask me. So, so what does it mean to read something? You know, like, well, what it means to me is, it means Horatio Heretius said, the author who will make me weep must first weep himself. You know, to read, for me, when I read something, I want to be able to be brought to tears, like Les Miserables or Kristen Lavransdatter can do. And, you know, I look at this particular text, which is one of the great texts in the history of science, in Farsi, in Armenian, and in Korean. I can't read a word of it, and it evokes nothing. It evokes no emotion. I have no idea what it's about. But when I see it in English, I see there's grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. That's how I want to understand the genome. So we have this great encyclopedia, the Genomic Encyclopedia. And it's true, you know, up there at the red tab, the protein coding parts, we know how to read that encyclopedia. But on the other parts, we suck. So, you know, we know the genetic code, you know, that's awesome. It's one of the great experiments. I'm truly overwhelmed by how cool it is, what Nirenberg pulled off. But for other parts of the genome, we literally know nothing. We literally know nothing. And I think that this point is made very well by this figure that Ernan made recently, which shows you the genomic ignorance in E. coli, which shows that for 65% of the genes, roughly, we know nothing about how they're regulated. That means that you find the gene, we don't know anything. We don't know where RNAP binds. We don't know if there's any transcription factor binding sites. We know nothing. On the bottom, oops, I forgot to put in the corrected version, but in Drosophila, it's so much worse. For 89% of the genes, we know nothing about how they're regulated. So, you know, to my mind, we may live in the information age of the genome, and we may live in the age where we can read and write genomes at will. But to my mind, it's pretty hard to write the poetry of genomes if you don't know how to understand the parts of the genome that tell genes when to be on and when to be off. So the point is, we really don't know much about the regulatory parts of genomes. So what I want to tell you about is an approach that really was formulated originally by Justin Kinney and his PhD at Princeton, and then was taken up primarily by Nathan Beliveau when he was a grad student in my group, and then repurposed by Bill and Susie in this really cool way that tries to use sequencing as a biophysical tool. So here's the idea. There's a particular promoter for which we know nothing, and we want to know how it works. And so the concept is to mutagenize that promoter, let's say at the 10% level, that is to say 1 out of every 10 bases in the promoter you mutagenize, and then you make a bunch of strains. You grow them up. You grow them in a bunch of different media, and then what you do is you find out how much expression there is for each of the different mutated promoters. And the thing about this, what's cool about it, is that because when you mutate a given letter, you may either increase or decrease the level of expression, you can actually get a map of meaning. And the way you might think of that is, you know, if I take the word walk in the English language and I mess with the letter A, you will still know that it's the word walk. But if I mess with the letter W, then you will not know that it's the word walk, because it could be balk. And I can play this game with other words as well. So the point is, the W carries more information than does the A. And so what we get out of this is what's called an information footprint, which is shown on the lower right at this point. The idea is that you see that certain bases actually matter a lot. And so this is a map of what matters, if you like. Then what we can do is we can construct a regulatory hypothesis. We can hypothesize that the region in red is a repressor binding site. The reason is because when we mutate it, actually the gene expression goes up. And we mutate an activator binding site that ruins the activator binding, and so the expression goes down. We can also determine what's called an energy matrix, which allows us to calculate how strongly the transcription factor binds. And then finally, using mass spec, for example, one can identify the transcription factor that binds at the site of interest. And so you start with ignorance, and at the bottom you end with a regulatory cartoon. So, you know, as I said, Nathan spent a lot of time working on this stuff, and this is an example of the information footprint for the well-known lac operon, and you basically can see where the CRP binds and where the lac repressor binds and so on. This is just to give you a flavor of how the mass spec part works. You take a cell, you break it open, and then you run the contents across an oligo that has the target putative binding site. And then using magnetic beads, those proteins that have bound to that putative binding site can be isolated and then subjected to mass spectrometry. And what you're looking for is enrichment of a certain protein that bound to this target site. And so there you go. So now I can come back to the puzzle. So what does this particular genomic sentence mean? So it's actually associated with a gene which is called PureT, and it's cool. It's related to purine synthesis. And the reason I like that is that it's a great gene because it has to do with the balancing of purine and pyrimidines. And this makes me think of another Austrian, another foul-tempered Austrian named Erwin CHARGAFF. And, you know, I don't know if the younger among you, if you even learn CHARGAFF's rules anymore, but they were very important to Watson and Crick. This is the rule that tells you the number of A's should be equal to the number of T's, the number of G's should be equal to the number of C's. And it was a biochemical discovery made by this highly refined Austrian gentleman who wrote this book called Heraclitean Fire. You know, he was like Schrödinger. He knew every language and, you know, quite an intellectual. He was really irritated that Watson and Crick come along. And, you know, he says, so far as I could make out, they wanted unencumbered by any knowledge of the chemistry involved to fit DNA into a helix. Well, sorry, they were right and you were wrong. You know, you don't need to know everything to do research. the way that Shaolao puts it, you just need to know something that's not known. And they did. So, so anyway, why do I like this gene so much? Because I like biological feedback. The cell knows when it's got too many purines or pyrimidines, and this gene is in on that act. And so using the REG-SEQ approach, back then it was sort-seq, but Nathan was able to identify that pure R actually binds here, and so on. And so, this gives you a sense of our before and after. And at this point, we've done a sort of a proof of principle of 100 genes. I'm not going to belabor this, but what comes out of that, first of all, what's shown on the left is intended to convey to you that there's a bunch of, we did a bunch of case studies where we already knew the answer. And the reason for that is we wanted to show that we could recover what, decades of labor of biochemists and geneticists and microbiologists have already discovered, that we can, basically find the same architectures using the REG-SEQ approach. And in general, it works. It doesn't work perfectly, but in general, it works. And then on the right, that's a series of discoveries, if you will. In other words, those were previously genes for which we knew nothing. And, to my mind, all of this is nothing more than a preface to our real job in my lab, which is,
Hey, Rob,
we want to try to predict. Yeah.
before you jump into this part, Kat has a question related to the REG-SEQ stuff,
Who?
which is
No.
Kat.
Sorry, Kat's not
No?
allowed to ask questions.
All right,
No.
so we'll
No, go for it.
Do you have thoughts on inferring promoter regulatory functionality beyond increasing and decreasing steady state levels, i.e. rate of production, timing, leakiness, etc.? How will we know when we've accounted for all the important aspects?
Yeah. Well, okay, so that's why this slide is here right now. And that is, as you know very well, Kat, I view the REG-SEQ thing as just getting the facts in place so that we can do, with the sorts of things that you've done with us. In fact, I mean, I think if I'm remembering correctly, Kat, you were in an experimental rotation in my lab, and that was when you were with Mason, I think. So, you've been down this path, right? And my answer is, well, first of all, we're never going to know that we got to the bottom of things, but, we have to, in my opinion, we have to suffer. We have to sit there and do the painstaking work. And sorry, my usual rant about surprise. We're not after surprise. We're just after getting it right. So, we've got to exactly as you said, examine leakiness, dynamic range, where the EC50 is, the Hill coefficient, all that stuff. We just got to do the hard work. And that's my answer. Hopefully that's good enough. So yeah, I mean, Kat's question was perfect. So, and maybe that's why Ernan asked right now. So yeah, we want to be able to try to make predictions about what all these distinct regulatory architectures do. Because I can tell you, we as a field have no clue, for example, about the role of multiplicity of binding sites. In other words, if I have 2 rather than 3 binding sites, or 3 rather than 4, for a particular transcription factor, we are not on top of that. We are not on top of binding site position. All these things remain enigmatic. So just to give you a little bit of a flavor, again, kind of in the spirit of Kat's question, of the kinds of things that we want to do is, we want to take the underlying architecture, once we've discovered it using REG-SEQ, for example, and then we want to predict what the outcome of a given architecture is. And we can use different methodologies. We can use chemical master equations, i.e. spread the butter. We can use equilibrium statistical mechanics, i.e. thermodynamic models. We can ask the question, when do they break down? We can work out the probability of binding and so on. And then you can make predictions, and again, I think a lot of you on the call, having been in the course, know that how this plays out. These are parameter-free predictions that I'm showing you here. And the ones on the top are not trivial. I mean, that's a log-log plot. And to get that kind of weird structure on a log-log plot is not obvious. It's probably one of the coolest things that's ever happened in my group, was that prediction and then the corresponding measurements. And, I think most people don't believe that we do the predictions first. But, at the end of the day, integrity is integrity. And I referred earlier to the river of time. Integrity is not a punctual thing. It's something about a career and about how you behave all the time. And, people can believe what they want or not. At the end of the day, the people that have been in my lab over this river of time know very well that, we made these predictions, and then these are the results. And then, you can argue about whether or not the theory works. Like in the lower right, we pain ourselves. And this is something Janne brought up the other day. We pain ourselves. Is it worth spending 3 years of a grad student's life to chase after the distinction between the green and blue dots and the green and the blue curves? I don't know the answer to that question. And maybe it's you guys, the youngsters that come into the lab that are the ones that will decide that for yourselves. So, we make these predictions, and I wanted to comment on hidden variables because that's also a very important part of this for me, finding the natural variables of a problem. So the other day when I introduced Maha, I was telling you about his advisor, Joe Keller, shown here. And again, I hope some of you had the chance to see him on MBL Beach. He wrote this great paper about the probability of a heads. And as I mentioned the other day, all you need to know is the initial upward velocity, because you know that the total travel time is 2 times the velocity initially upwards divided by g. And then you need to know the initial rotational velocity. And so from those 2 numbers, you can construct a dimensionless ratio. It tells you whether you get a heads or a tails. And it's a hidden variable. Like if I happen to know those 2 parameters and you didn't, I could make predictions about what's going to happen to the coin, and you can't. And the reason I bring it up is that, a few of you that have had the fun or the punishment of working with us at MBL have done what's on the right-hand side of these graphs, which is finding the natural variables for biological problems, and then working out the corresponding data collapse. And when I say data collapse, what I mean is not a fit. It just means finding the variable that the cell cares about and not the pipetter's variables, which are the things we care about. The cell doesn't care about our pipetting of the inducer concentration. What it cares about is a weird combination of number of repressors, strength of binding sites, how much inducers around, and what mutations are present. But when you know all those things, you can figure out the underlying free energy. And for those of you that might be interested, Griffin Chure has a really nice paper. Sorry, that lacks humility to say it that way, but I didn't do it. He did. In PNAS from a year or 2 ago that really talks about this. So that's the end of my part on the genome. And what I want to say is just imagine what we could do if we knew the rules of writing the poetry of the genome. So that's what I have to say on that part. So now what I want to do is I want to turn to chapter 6 of Schrödinger's book, which is where he talks about order from order. So, so he was interested in the emergence of biological order, and he wanted to argue that it was, from the standpoint of statistical physics, a different thing than order from disorder. So what did he mean by order from disorder? This is actually, the figure on the left top is actually from What is Life? It's kind of funny because it looks very much like the sort of thing you'd get from a Gillespie simulation of diffusing particles, for example, if you had a source and you had degradation. So what he had in mind was this, that he had in mind the statistical structure of classical physics, the Boltzmann physics, and really it's so deep and simple all at once. And the idea is, it almost sounds like a tautology. The system will evolve towards its most likely state. That's it, and what's plotted here is the entropy when you have a wall separating 2 different reservoirs, and then you allow them to now start communicating. And what happens is that the entropy of the reservoir on the left goes down, the entropy on the reservoir on the right goes up, but the total entropy is maximized at the point in the middle where you've basically got them equally distributed. And that corresponds to equality of chemical potential. So equality of chemical potential is really nothing more than maximization of entropy. So, so he makes a lot of noise about the diffusion equation. And in fact, he writes it down. He writes down the partial differential equation that I show you here on the left. And, I think it's really great because what he wants to say, and he does it so well, he says, listen, every molecule is doing nothing more than flipping a coin. He says it literally in those words. Every molecule is just flipping coins. And from that disorderliness, you reveal what I show you on the right, which is basically the incredible orderliness, the green function for the diffusion equation. It couldn't be more orderly than that. And yet it's nothing more than the emerging from the laws of disorder, this second law thing that I wrote down a minute ago. So he's very interested in what's different about lifeiness. How is it that living organisms don't run down sort of to equilibrium? And he really, I think, he's got many messages. He's got messages about the role of quantum thinking in living matter and also about statistical physics. But at the end of the day, I think it's more important to say, really what he's telling us at the end is that we don't know what the physics is yet. We're going to find it out, and it's going to look just like all the other kinds of laws we're used to. That's his speculation, which I happen to share, but we don't know it yet. So so the reason for this slide was just to say, I think our failure to be on top of non-equilibrium physics is a failing in physics that hurts biology. And biology demands of physics now that it gets its house in order on the subject of non-equilibrium. Saying non-equilibrium physics is like saying non-elephant biology. So, what's up with this order from order thing? And, just like I said earlier, let's not forget the vitalists. They were not stupid. Lamarck wasn't stupid. They were just acknowledging that somehow the stuff of life is weird. It's doing weird stuff. And, if we take a sort of modern point of view, I like this figure that we've made. It's all the ways in which, biological systems, or not all, a bunch of ways in which biological systems, by expending energy, exactly as Jeremy told us at the beginning, that they can do things that equilibrium defies. You can create concentration gradients. You can do high fidelity polymerization, very low error rate translation and replication. The very amazing fact that biological systems adapt, chemotaxis or vision, it doesn't matter, they adapt. That costs energy. It costs energy to be on top of that. What Julie likes to call socks before shoes kinetics, the kinetics in which A happens before B. One-way arrows, the way that Jeremy Gunnar-Wardena puts it in his paper on Michaelis-Menten, which I really think all of you should read, is that's the original thermodynamic sin, is drawing one-way arrows. And you see it all the time in biology books and in papers. You're not allowed to have one-way arrows. They violate physics in some sense. So building self-organized dissipative structures, doing post-translational modifications, and then ordinary things like doing work. So this vignette is kind of inspired by the idea of accounting for biological order, as Schrödinger had in mind. At least that's my interpretation. And what I want to tell you about is some work that basically was done by Tyler Ross with Matt Thompson. Matt and Tyler bravely undertook this while Matt was like a super postdoc at UCSF. And then when Matt moved to Caltech, Hun-Jin and Rachel joined in on the fun, and they did this. The four of them did this amazing stuff along with Ziji also. So the idea is basically to activate the cross-linking of motors in space and time as they see fit. Really, what I'm saying is optogenetics. You use light in order to cross-link these motors. What that does is to dimerize these motors, and that actually cross-links the microtubules. And what I'm going to show you here is a video of what happens in that disk of light, which is that you basically form an aster, the light is turned off, and then this thing goes away. Here's several other examples, that they were very inventive as they thought about this. So the one in the middle is actually inspired by what Julie told us was her favorite all-time movie, which was the neutrophil chasing a bacterium. Here, light is being used to basically move these asters around. So the asters are following the light. The one on the right is just to show you the interesting flow patterns that are induced by these things. And what Rachel and Hun-Jin are up to at the moment is using different motors. So, we're interested in knobs. Very interested. This goes back to Kat's question. What do we want to do in my lab? I want to tune the knobs as a theorist and as an experimentalist. So the knobs that we might tune are the speed of the motors. We might tune their processivity, and then ask questions such as how big are the asters as a function of processivity and velocity. And this is just to give you an indication that Rachel has actually already made lots of progress on this. This is 3 different motors, so 3 different kinesins, and the corresponding diameters of the asters, all with the same diameter of illumination region, which I think might have been 600 microns, but I forgot. So I apologize for that. And then Vahe has actually been working very hard to, and many of you will know Vahe from MBL. He's been working very hard to write down field theories that allow us to try to make contact with those experiments. So, the idea is exactly what you might have imagined from the PBOC course, which is, we define the relevant field variables, which is the bound and the free populations of the motors. And then we write down some PDEs, basically reaction diffusion equations that describe the behavior. And then we can solve for the motor profile. And Rachel has been doing tons of different measurements, and Vahe has been actually looking at the distribution of microtubules and the distribution of motors. And so, again, that's just to give you a flavor of what the word accounting for, or the words accounting for, mean to me in the context of this active matter problem. So I'm just going to spend a few minutes on what I think we might have learned, I guess. So, so the first thing I want to say has to do with the status of laws and principles in biology. And, what I would tell you, and, I often work from frustration, and, I've been very frustrated by the defeatism that I've encountered in my 20 years since I've shifted into biology. And I'm quite honestly, most of you who know me well will know I'm incredibly tired of it. I'm tired of people telling me it's too complicated. You can't model it because it's the product of evolution. Give me a break. So careful physical analysis contradicts this defeatism at nearly every turn. And, three of my intellectual heroes are Phil Nelson, and I hope he's here, but if he's not, he still is, Bill Bialek and Ray Goldstein. And, all three of these people have written deep thoughts about the status of law in biology. And I love what Bill had to say. And these could have been perfectly coming out of Schrödinger's mouth. He says, we're trying to understand how the same forces that usually cause carbon-based materials to look like rocks or sludge can, under some conditions, cause some of this material to organize itself and walk or swim or fly out of the laboratory. What is special about the state of matter that we call life? How does it come to be this way? Different generations of physicists have approached these mysteries in different ways. It makes me think of Perutz. Give me a break. He can't tell me how those special states of matter walk or swim or fly out of the laboratory. Not even close. So this is one of my favorite examples. It's like probably my dream problem, and probably that means I'm gonna work on it. I'm just gonna let you watch. I'm gonna sit here quietly. Well, I could sit and watch that all day. That makes me think of Heinrich Hertz and contact mechanics. So Hertz, was one of the great physicists. He was Helmholtz's student. He died so young. He didn't even make it to age 40. And, probably most of us know him for his experiments, which is funny because he was a theorist, but he's the one who basically confirmed Maxwell's theory. He discovered electromagnetic waves. He measured them. But he also did a solution for what's called contact mechanics, which has been incredibly important. And, it's kind of a funny aside. I'm showing you here on the right a paper by Margaret Gardell and Uli Schwartz and Claire Waterman that I love. It's one of my favorite papers. And, what I love about it is shown in the upper right, which is that's the elastic green function for a half space. What's a green function? It's the displacement field that you get from a point load. And they use this green function actually to develop traction force microscopy. And that ultimately traces back to Hertz. So Hertz figured out in like 1882 or something, how to understand the elastic deformation of a material that you push on. And this thing is an indentation problem. The sea lion is indenting the school of fish. And yet, this thing has nothing to do with Hertzian contact mechanics. And I've actually been doing a lot of experiments myself on this. And it's very interesting. Like you can see, the fish, they only will displace a little bit unless somehow this instability starts. And over and over again, I've taken videos and tried to understand, you poke at them, and they will move. And actually, I've noticed the same things with coyotes and mountain lions. You poke them, meaning you perturb them, but there's a weird sort of thing going on. So what did Schrödinger mean by new laws? And I guess I want to say we don't have to be grandiose. There's laws with a little l, and then there's laws with a capital L. And there's tons of laws in physics with a lowercase l. Hooke's law is an example. But so is Fick's law and Ohm's law and Fourier's law, et cetera. Like those are provisional. Ohm's law is not some universal thing. It's just provisional. It's a linear model of what happens when you do a little, weak perturbations. And, on the right, this is a figure that we made for the new edition of our book. And, it makes me think of Yane because, it's all the different ways of spreading the butter, which I think is kind of funny. And, there's lots of opportunities for that kind of small scale law making in the biological setting. And I wanted to say, there's this great talk by Cavagna, which I would recommend you watch, about flocking. It's called The Seventh Starling. And it basically explains to you that although for 2,000 years, people have been watching the starlings in Rome, it was only in the last 15 years when they put up cameras and did the kind of image analysis that all of you learned to do at MBL, that they actually were able to discover new laws of active matter, and the force of interaction between the birds is not like a normal law, so that's cool. And, I'm obviously a big fan of ASTER, as many of you know, and, a total Ned Wingreen groupie. And he had these two papers, one on chemotaxis and one on quorum sensing, in which he showed this data collapse. And then on the right is the thing that I showed you earlier from the socialists in my group. And, all of them show you there's a lawfulness to ASTER, the Monod-Weyman-Changé type model is really interesting. So, so I guess I'm close to done. So Schrödinger, in my opinion, what's this book about? I've been having this argument with a friend of mine who I met in physiology. She's a professor at UCLA now. And we've been having this argument about, is it about quantum or is it about statistical physics? And I don't think it's about either. I think it's about raising our standards. That's what Schrödinger's book is about. It's a plea to raise our standards. And he says, as he says at the bottom, there's just one general conclusion to be obtained from it. And that, I confess, was my only motive for writing this book. It emerges that living matter, while not eluding the laws of physics as established up to date, is likely to involve other laws of physics hitherto unknown, which, however, once they have been revealed, will form just as integral a part of this science as the former. So I wanted to make sure that I thank David Booth, who is the one a couple of years ago who suggested to me that I write an article on the 75th anniversary of Schrödinger's What is Life, which I've done, and it's almost done, and I'm going to put it on the archive probably in the next 2 weeks. And also Carl Zimmer, because he and I have had like this amazing 6-month exchange. He's writing a new book on what is life that is really interesting and gave me, for example, the story of Hydra. And when it comes out, you all must read it because it's full of stuff that you will not know. And it will remind you that you're part of the river of time. And hopefully it will remind you that we need to have humility because there's so much confusion along the way. And I think we're still in the middle of it. So, this question he asked, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? And, I think, the point of our course is let's not lose our sense of wonder and the mystery of the world, and the way I think of it is best illustrated by a quote from Thomas Paine, which was in Common Sense, I think. A long habit of not thinking a thing wrong gives it a superficial appearance of being right. A long habit of taking things for granted gives you the wrong impression that they're understood, but they're not. They're totally not. And so, accounting for a la Schrödinger, it's not been done yet. Now is the time. So a few more words of gratitude, and then I'm going to end with a few words about the magic of MBL. So, my two author teams, I've learned more from them and had more fun with them than, basically anybody in my scientific career. And that's true to this day. So, in some sense, I dedicate this talk to all of you guys. And, my group members, it's amazing to have worked with Nathan and Susie and Bill and, Griffin, Manuel, the socialist, Stephanie. And, I'm probably missing people, and for that I apologize. And then, this new adventure in active matter has just been, an amazing experience. So, that started in physiology with Christina and Sophie and Vahe, and then Tyler and Matt. That was amazing, and Rachel and Hanjin got involved. So I just want to finish with the magic of MBL. So what is that MBL magic, and who are the magicians? Why is it that it feels terrible not to go there? So you guys that at least have been in the course know about the lightning talks. And, I always laugh at the lightning talks on the first day, and I always cry at the lightning talks on the last day. And, it's even hard for me to get through this right now, but I have permission from the two people. I'm not going to tell you who they are, but the two people for whom these are the relevant slides. So this is the lightning talk from, I think, 2015. MBL from somebody I'm very close to. And I love this, you know, you think science maybe or life or whatever is about the destination, but in fact, you know, it's the journey. And of course, this particular picture means a lot to me. And I know JLS is here. So, you know, she and I swam across there to the dock. I don't know. I think I estimated, I did it like 780 times over the last 10 years or something, but she and I did it quite a few times. And then this is somebody I'm hoping is on the call. I'm not sure about this, but it's really funny. So, you know, we're talking about on the left, this is my antisocial grad student. I have ideas, but worry I'm not up for this. And then, you know, the MBL with its incredibly intellectually stimulating supportive environment, ridiculous amplifier plus awesome filter. Wow, is he always as talkative and excited? I love that. So, you know, Wallace is a great guy to hang out with, and he's such a nice picture of him. He looks so happy, and yeah, but you know, he's kind of always on the edge of rage, actually. And so, in talking about MBL, he said this: skeptics have often asked me, what is the unique aspect of the MBL that makes you want to be there? And after years of consideration, I've come to the conclusion that there's in fact no specific unique physical factor. What makes people want to spend their summers at the MBL is the fact that people spend their summers at the MBL. The metaphor, et Thomas, think about this. The metaphor I like to cite is the French village of Provins, which became one of the most influential and important cities in medieval Europe, despite having absolutely not one single unique advantage. It was not located on trade routes. It had no ports, had no special natural resources, no great defensive positions, no special agriculture. What set that place apart was its convening power. The local prince or whoever he was had the idea to host a trade fair in his town, and gradually it grew so that people would come from all over Europe to buy and sell things like spices, furs. Well, actually, I have no idea, but whatever it was that people traded in the Middle Ages. Can't you guys that know Wallace, can't you just hear him saying this? Swords, I guess, and that sort of thing. Anyway, the point is that people came to Province every year when the trade fair was taking place because they knew that everyone else was going to go there. It was all about convening power. And so, you know, that's the magic. So I'm going to let Dyke.
Here's a test. If you see anyone who has taken a course at the MBL or done summer research here, taken a physiology course, say, just mention the word MBL to them or the word Woods Hole and see if they don't get kind of a misty, faraway look in their eyes and start talking about the magic of the place. They'll start to sound a little bit like maybe some sort of religious fanatic talking about a cult, maybe. If you haven't been here, that might be what they sound like. But if you have been here, it will sound more like an expatriate talking about their homeland, you know, a place where they feel some deep connection to what they're doing.
There you go. Well, that was weird. I mean, the lack of questions is always a bad sign, but that's what I got. I'm early, which is what I like to be.
All right. No, there are a few questions, just that there were on things that you had already passed, so I thought we would just leave them for later. So there's one from Shri about the natural variables. Do the natural variables in this specific example provide insights about what details don't matter? For example, details of underlying architecture or dimensionless combinations of parameters?
Absolutely. Yeah. I mean, it's, in fact, good morning, Sri. It's so nice. It's been nice to see you every day. It's so incredibly cool. I have to tell you, you know, again, maybe if you, well, let me, I'm going to make an offer, which is I'm going to offer that Griffin is going to get in touch with you and he's going to send you a few slides because there's an equation, which I should have written down. And, you know, it's a nonlinear function of the concentration of repressors, of the binding site strengths, of the inducer concentration, and it's something you never would guess. And yet, when you know it, you can use it with incredible power. And what I mean by that is that you actually can make predictions about quantities that you wouldn't even have dreamed of. And so maybe the easiest way to say it in this particular case, and I think you will see this, is that the level of gene expression basically depends on how the probability of occupancy of the binding site by the repressor. There's many ways that I can get to the same value of that occupancy. I could turn up or down the amount of inducer, I could change the number of repressors, or I could change the binding site strengths. All three of those can arrive you at the same occupancy. And in so doing, you get the same level of gene expression. So what's shown on the x-axis here is basically, if you like, is the occupancy. That's really, in some sense, that's not what you're plotting exactly, but that's what you're tuning, is the occupancy. So you get to the same occupancy from many different routes, and that's what the natural variable sort of characterizes. And actually, now that you mention it, and I'm so glad you're the one who asked the question, I had the pleasure of sitting in the audience in Santa Barbara, and also you came and talked to my group, and you do exactly this yourself with respect to your growth data, right? I mean, you've found the natural variables for the growth in Colobacter. So, it's a very similar thing, although probably, maybe you won't agree with this, but probably we're more on top of the underlying theory in our case than you guys are, because there's a particular molecular thing going on, which is the binding of transcription factors to DNA. So it's easier.
So related to this, and you kind of touched upon it already, but could you explain the collapse of data? I didn't exactly understand what you meant. I think maybe the power of data collapse, what it teaches you.
Yeah. Let's see. Well, let's see if I can figure this out. So just bear with me. Can you guys see my screen?
Yep.
Okay. It's a little scary, but I guess I'll do it. Shoot. Does anyone see a LaTeX file? Oh my gosh. This is getting embarrassing pretty fast. All right, well, okay, good. So, I want to show you something because I'm hoping that it will show you what this data collapse thing is all about. So, oh, I skipped it. Yikes. Oh man. Okay, good. So can you see this, Ernan? Is it visible?
Yep.
Okay. So up here in the top figure, I'm showing you 3 binding curves. So this is, all you need to imagine is the equation C over KD over 1 plus C over KD, the Langmuir binding curve. So every receptor will have its own special curve, but that's because I'm not using the natural variables of this problem. The natural variables of the problem are C divided by KD. If you use those variables, all the curves collapse as shown here on the right, okay? In other words, the variable so when I say data collapse, what I mean is that you think you have many different curves, but you don't. They are really a 1-parameter family. It's just that you have to figure out what the variable is that the system cares about. So the second one that I'm showing you, figure 317, that's a very classic one, which is the so-called law of corresponding states. And if I were to plot these in non-dimensionless variables, every material, neon, argon, krypton, xenon, et cetera, would have its own curve. But if you plot the temperature divided by the critical temperature and you plot the density divided by the critical density, they all fall on the same curve. In other words, there's no fit. You just plot the data. That's it. It's really that simple. And we've done this in the context of ion channels, so this is data for a bunch of different ion channel mutants. And what's shown on the right is if you know the natural parameter, they all collapse together. So hopefully that was a reasonable answer.
Thanks. Carolina?
Thanks for the great talk, Rob. I have to say, it made me feel a little bit like if I were in Woods Hole. So that was greatly appreciated. One question that I have from you is a more philosophical question. So you mentioned that we don't understand these systems yet, and these problems are not solved yet, which implies that we will get there to solve them. I wanted you to speculate a little bit more about that. If we'll actually ever get there, how much we'll be able to understand, and what are chaotic systems?
Yeah. I mean, you've probably heard me say this before, and I sure as hell believe it, which is the following. When somebody my age tells you something can't be done, you should tell them to stick it up their ass, just ignore them. But if they tell you that it can be done, you should listen to them. I mean, where is there a theorem that says you can't know stuff? Where is there an example from history of us really failing to I don't know. Sorry, that was uncalled for, like hostility, but I just get tired of being around people that are always saying you can't do stuff. So over and over and over again, we've been confused, and then we've figured stuff out. So I mean, I guess I just feel like I keep on seeing examples when people pay the price and dig hard on these problems that they can understand them. So does that mean that there's a grand unified theory of everything or something? That's not what I'm implying. That's why I guess I was keen on the law with the lowercase l. And, you know, I don't know if you've taught from our book yet or if you're going to, but, you know, in chapter 1, we have this whole little thing about, you know, the different ways of viewing DNA or the different ways of viewing membranes. And the reason for that is that, you know, depending on your problem, you might want to view DNA as an elastic rod, but sometimes you might want to view it as a random walk. But when you view it as a random walk, obviously that's not going to tell you anything about how to think about it as a genome that's doing transcription, you know, or not much. So I don't know if I'm really answering your question other than saying that the objective for me, you know, this is subjective, but of science is to understand stuff. And history has taught us that science is the greatest engine of knowledge generation that we have. And even though there are examples like weather that continue to be challenging, you know, we've gotten even better at that. And it's a chaotic system to the extent that I understand it. So I don't know, are you still there, Carolina? I mean, maybe let's have a little bit of an exchange since we still have a little time left. Why don't you?
The Zoom exchanges are always harder, but no, I agree with you. I mostly wanted to have like that the usual Rob positive note. It's always good to end on that.
Well, sorry, and yeah, but then I had to plug into my Wallace like rage too.
Yeah, I love when you read the quote from him. I could totally hear him speak.
Exactly, right? Isn't that funny? All right, what else you got for me, Ernan?
So Gabe has a question. So various attempts on Sanger Esposito, Gabe Salmon.
Yeah.
So various attempts on Sanger Esposito, Crooks, Jarzynski have emerged to wrestle with non-equilibrium over the decades. Can you comment more concretely on the specific ways in which these existing earlier attempts at non-equilibrium are inadequate? What would more effective a more effective theory look like or accomplish?
Yeah. So for those
are you
of you
dissatisfied,
who don't,
I guess.
Yeah, for those of you who don't know, Gabe is my a grad student in my group, and you know, like this is the quintessential advisor moment. It's like, dude, that's your job right there. I mean, you just wrote the abstract for your thesis to answer that question, and that's literally what I want you to do, frankly. Look, I am very excited about what Jarzynski and Crooks and Jeremy England and many others have been up to. Okay, that's point one. I'm very practically minded. Okay, I'm super not interested in the philosophy from Prigogine and other people like that. I want to solve real problems like Rayleigh-Bénard convection, the Jupiter red spot, you know, the active matter things. And I guess, you know, I feel like we will know it when we see it. And you know this very well, Gabe, but the bottom line is that in equilibrium, we have one simple principle that presides over everything. It's mathematical, it's variational, it comes from Gibbs. Now, I have asked a lot of people over the years whether they think such a thing can or will exist for non-equilibrium. And this goes back to Carolina's question too. And many, I would even say most of the sophisticated theorists that I know think it can't be done. I'm personally, you know, I'm optimistic. I think that they're guilty of, because I haven't done it, it can't be done. If you were in the year 1565, there's no way you could have figured out Kepler's laws. No way. You needed Tycho Brahe. So I feel like, you know, the push for precision measurement and, you know, little steps for little feet is what's needed. And also designing the experiments that are the dialogue between those theories and experiment, concrete, you know, pragmatic, detailed. So anyway, you shall pay for that question. I'm just kidding.
Thomas?
What?
Did you? No, I don't know if Thomas wanted to say something. Oh, okay. Okay. So one question that this is now going a little more into specifics. The notion that biological equations should have a backward arrow seem a bit confusing to me. Let's say a signaling cascade occurs and transcription results. How can this be reversible?
Yeah. Yeah, no, I like that question, but at the end of the day, you know, there's still a rate for the chemistry that happened to go in the backward direction. There is. I mean, you know, like if you think about the notion of the well, okay, so there's a few pieces of this that are super interesting. So one point is that by the time we write down the kind, you know, I don't know who asked that question, so I don't know where you're coming from, but like if you've been with us before at the whiteboard at MBL and you've written down models of transcription, notice the level of coarse graining. You know, like we write an equation which relates a state with no polymerase and then a state with polymerase to a rate of production R. You know, there's a lot of stuff that's ignored there. So, you know, really I should be talking about the transition from no base pairs of the messenger RNA transcribed to one base pair, you know, from one base pair to two and so on. And then there will definitely be reverse arrows. But you ask a very deep question at the level of like the coarse grained models, you know, and in that sense, I'm not sure I really have an answer for you because we definitely do all the time write down one-way arrows in those kinds of models. But, you know, at the end of the day, and please read Jeremy's paper and, you know, you can send me a mail and we can chat about it further. I'm not claiming that I'm on top of this. You know, it's also kind of related to Gabe's question. We don't know how to coarse grain non-equilibrium systems very well, or at least I don't.
All right. So from Boyan, a very general question that I would like to hear your comment on. There are two ways to study something. One is to observe a real system and to try to understand and gain knowledge. The other is to try to build something using what we know, like you did with the optical genetic motor assembly. You know, this is going back to the what I cannot create, I do not understand. What are the ups and downs of the two ways? Which is better, even?
Yeah, I don't know if I'm game to say one's better, but I am game to say that in my opinion, the forward progress of biology depends on self-conscious, detailed, aggressive versions of the second thing that you said. What I mean by that is saying, you know, fine, I'm not talking about the quote unquote real system. Doesn't matter. I'm going to try and figure out what the principles are of this thing. And, you know, there's an example that Ernan and I love, which I'll show you. So let's see. This is the example of the quantum corral. So, you know, like if you study quantum physics, which probably you did actually, and you learn how to solve the Schrodinger equation, one of the first things you'll do is the particle in a box. And so, you know, usually you'll do like the one-dimensional particle in a box, but then the next one you should do is the two-dimensional particle in a box, which is in a circular box, for example. And so, you know, this is as old as quantum mechanics. People have been thinking about that forever. But it turned out in the late 1980s, early 90s, Don Eigler at IBM figured out how to make a 2D quantum corral, and then you could actually test out your understanding. And, you know, this diagram right here shows you the energy levels that you get out of increasingly sophisticated treatments of this sort of problem. And, you know, nobody would claim that the ones on the left are real in any sense. And yet you learn how to handle your craft on those unreal problems so that when you come to the real problems, you actually know what to do. So I don't know. I mean, the bottom line is that, you know, my strong reaction to your question is because there's so much emphasis on that's not the, you know, like just think about the hierarchy of criticisms in reviews where, well, that's not the real system. That's, you know, even if you're doing tissue culture, then that's not the real system. And then if you do something else, that's not the real system. And everybody's always telling someone else, well, that's not the real system. Sure, fine. So what? You know, so I guess my answer is I'm a fan of both. Okay.
All right. There's a question from Sechen, and this is, I think, you know, touching on to probably the Bialik board view that you mentioned during your talk. Isn't there a missing part of Schrödinger's question, which is what functional good does the physical process happening in the cell do to its survival? Example, when can we predict the fold change accurately? In what sense is it useful to talk about its relation to some kind of optimization or fitness?
Yeah. I don't know. I mean, that's for you to find out. You're like 23 years old, so I mean, that's a great question. You know, little steps for little feet, I'm afraid, is my take on that. It's a great question. You know, at the end of the day, there are questions that you're ready to answer and there are questions you're not ready to answer at a given time. And I think we're entering the time period, Session. I mean, you know this because you know people in my lab are now trying to bring evolution into the fold and make it a biophysical thing. And, you know, there are many other people that are up to that in really interesting ways. And who knows, maybe you're going to join one of those groups at Harvard. I don't know, or maybe you did already. So the bottom line is that, you know, once again, you got to do your spade work. It goes back to my response to Kat. You've got to go out there in the world and you've got to do your hard work of digging into the details and seeing whether or not there is a functional consequence. You know, look, I mean, let's just remember, Schrödinger gave 4 public lectures in Dublin. That's the origin of his book, public lectures. I just want to emphasize that, 4 public lectures. So, you know, he was trying to get people excited about the idea that physics might enjoy contemplating that part of the universe, which we call living.
All right, I think we're out of time. So yeah, thanks everybody for being with us all these last 3 weeks. And thanks, Rob, for a great talk.
Great to see everybody.
Yeah. It's a little bit anticlimactic, but I guess we'll just hit, you know, end webinar, and that will be the end of it.
All right.
The best part about Zoom meetings is that you end the talk and then you turn off Zoom and then you're alone in your room.
I know.
That's true. That's very true.
Bad. Yeah. Be
Well,
nice
miss
to
you
hear.
all, and hope to see you next year, if not sooner.
Yeah.
Yeah.
Hope to see you soon.
Great. I wish we were
guys.
going swimming.
After talk.
Jennifer.
Yeah,
I wish we were going swimming this afternoon.
I know.
You know.
That really, oh, wow. It would be great.
Yeah.
Oh, well.
It would have been fun for all of us on this call to go sink the dock this afternoon.
I know. I know.
Yeah. All right. Well, it's great seeing everybody. Thanks for participating over the last few weeks. Ciao.
Thank you.
Great